Convergent evolution, wolves and thylacines. Let's break this down a bit. Convergent evolution is most commonly known as the evolution of similar features in independent evolutionary lineages. The process of convergence between species often requires a specific mechanism. These include differences in developmental processes that produce similar structures or an adaptation in response to the same selective pressures. Convergence is generally seen as the ability natural selection has in producing optimal solutions to problems in similar environments. The thylacine, also known as the Tasmanian tiger, was the largest known marsupial carnivore. The distinguishing features were the 15 to 20 dark stripes that ran vertically down across the back. Unfortunately, this animal has been presumed extinct since 1982 from significant threats including human, disease, and competition. The grey wolf was the world's most widely distributed mammal carnivore, living in the northern hemisphere. The animal is distinguished by its larger size and less pointed ears and muzzle. Their present distribution is more restricted, only occurring in wilderness and remote areas. Fortunately, the wolf population is stable and has been assessed as least concerned due to its relatively widespread range. Recently, there has been a focus on genetic basis of convergent evolution. The study of Thylacine genome has enabled a more comprehensive understanding in the genetic basis of adaptive phenotypic convergence. As previously mentioned, the thylacine was extinct, and the genome could only be sequenced from a preserved specimen. This means there was chance for error, but comparisons and methods were taken to minimize them. The research demonstrated the possibility of using genome-wide presence and absence patterns of retroposons to clarify the phylogenetic relationship of an extinct species. This allows other specimens to also have their genome sequenced and phylogenetic relationships clarified, even if the species no longer exist. We are able to reassess species in their phylogenetic relationships. Cranial comparison using 30 landmarks also revealed the initial convergence between canids and thylacines. Diet is a significant predictor of cranial shape. This reinforces how convergence can be seen through phenotypic resemblance. Additional research uncovered that the ancestral phenotype space between Tasmanian tiger and grey wolf is 40%, and a third of ancestral phylomorphospace has been closed by convergent evolution. These species have been separated for over 160 million years, making it an exceptional model to be used in future studies of convergent evolution-related species. The model can be used for teaching the concept of convergent evolution in school, or as a platform for scientists to discover other convergent evolutions in nature. Detection of convergent evolution at the amino acid sequence level has been around for a while, but is insufficient in identifying convergent changes. This method has mixed results and mostly depends on detection methods and convergent substitutions. Genome-wide studies attempt to identify homoplasious amino acids in protein-coding genes between convergent and non-convergent evolutions. We learned that protein-coding genes are not major drivers of convergent evolution, as selection on genes is limited and consistent in neutral processes found between thylacine and canids. This finding enables other scientists to research other genomic regions that could drive phenotypic convergence. Convergent evolution has been understood to be species with similar traits that have evolved independently. Studies on the genome of thylacines show that protein-coded genes in the genome are not major drivers of convergent evolution. This research is important, especially to evolutionary biologists, as it is evidence for the role natural selection has in evolution. When independent species evolve the same phenotype in similar environments, it would suggest that natural selection favors such phenotypic variation over others. Consequently, the importance of convergence at the genetic level remains highly unknown. Therefore, us scientists need to uncover such mysteries. I think it could give insight into understanding how the genetic background determines the change of complex traits, the constraints of molecular evolution, and an opening of a new field in genetics.